Hi everyone, welcome back. Here's our lecture for chapter 9 about the human visual system. We're also going to introduce the idea of sensation and perception. You can go on and take classes dedicated to this topic of the second midterm, sensation and perception in the department. So we're going to talk about 5 topics today. First, this general introduction of sensation and perception. Then we'll talk about the functional anatomy of the visual system. We'll talk about how the visual system encodes for different locations in our world. And then we'll look at the neurons' activity specifically and look at how the visual system encodes different features of what we see. And finally, we'll think about high-level vision, about how the brain uses vision to identify objects and act in the world. So our brain, in the terms of our vision, only receives input. All it gets is a series of action potentials coming from the neurons of our eyes, in this case, into the brain. And this happens the same in various sensory pathways like our auditory system or our somatosensory system as well. And we understand this idea of transduction, how nerves can transduce or turn energy like light waves into nerve impulses. And we also know the pathways that the nervous system sends this information into the brain. So for instance, we know the pathways from our eyeballs back into our visual system in the back of the brain. And what we don't understand, one of the remaining mysteries of the brain that's open to be solved, is how we perceive, how we subjectively feel one group of action potentials to feel like one thing, say the color red, and another group of action potentials in a different area of the brain to feel like something else, like a warm shower. And so even though those are both just physical action potentials, there's a subjective perceptual feeling that differs between them, and we know less about why that subjective feeling might be different and how we might learn about these differences. A sensory receptor is a specialized type of cell in the human body that transduces or converts sensory energy like light or sound waves into neural activity. Each of our sensory systems is designed to respond to only a narrow band of the world, a narrow band of the world's energy. So our visual system only has this narrow range of electromagnetic energy that activates the receptors in the back of the eye and turns that light energy into chemical energy inside the cells. Our auditory sensory system detects air pressure, and that produces a mechanical change in the cell that creates a cascade leading to the action potentials. And somatosensory system measures mechanical energy like impacts on our skin, and the taste and olfactory systems measure chemical molecules, as we'll talk about in the third midterm, actually, in the motivation and emotion chapter. When we talk about receptive fields throughout the course, we're talking about the specific part of the world to which a given cell, in this case a sensory receptor, responds. So the receptive field of a sensory receptor in your eye is any part of the world in front of you that when a light appears there, it makes that neuron in your eye, in your retina, fire. And these receptive fields allow, for instance, the visual system to always be sampling for information out in the world, and then also to keep a spatial representation of where that information is, because different cells have different receptive fields that are partially overlapping. So each photoreceptor in the back of our eyeball has a unique part of the world from which photons can arrive, the world with respect to your head, from which photons can arrive that will hit that photoreceptor and make it fire. So everything to the right of you, for instance, is going to hit the left side of each of your eyeballs, and everything to the left of you is going to hit the right side of each of your eyeballs. So a neuron on the right side of your retina, I just realized that the image may be flipped for you, but the neuron on the right side of your retina has a receptive field somewhere in the left visual field. In terms of sensation, the density of our sensory receptors is an important feature that determines how sensitive that given receptor area is. So we have more tactile receptors on our fingertips, for example, and we have more visual receptors in the fovea of our eye we'll see today. And these differences also can explain some of the abilities we see in our animal relatives that give them what seem to us like superhuman abilities, like the olfactory ability of dogs. They just have much more receptor density in their nose. A neural relay describes the fact that all receptors connect up to the cortex through a sequence of neurons. So there's a sequence of connections from the sensory receptor up into the brain. Sometimes 3 or 4 intervening neurons in between. And at each of these stages in the process, some information can be modified, some computation can happen on the information. And so the information coming in can start to be modified. So an incoming sensory signal, like a flash of light, can be different. It can be red or green, it can be a line, or it can be a line shaped in a different direction. And this incoming sensory information is encoded by only action potentials. The only signal that a neuron can send to the next neuron is action potentials. And in the first case of these sensory signals, they're traveling along peripheral nerves outside of our central nervous system and then going into our central nervous system. They're afferent. But how these action potentials encode different types of sensations like vision and touch is a matter that we're going to study in this class. And further, how might these action potentials, just different trains of action potentials, encode features of a particular sensation like purple or blue, differences in color within vision. We'll see throughout the day today and throughout the lectures this month that how the brain can encode for these different types of features of our sensation. The very most simple way that a stimulus can be encoded is that its presence can be represented by a neuron by an increase or decrease in the action potential rate. So action potentials can occur every couple hundred milliseconds, and this will happen faster and faster if there's a stimulus that's present in the receptive field of that neuron. Further, it could also decrease the firing rate of that neuron, and that would equally be a way to represent the presence of that stimulus. Right, so an increase or decrease in action potential is one signal that the brain can use to indicate the presence or absence of something. So how would these action potentials, however, encode various types of sensations? So this is a more complex question because different sensations, one answer is that different sensations, like our visual and auditory sensations, are encoded in different parts of our brain, our visual at the back and our auditory in the temporal lobe here. And we learn to distinguish the senses through our experience. We hear something over and over and over again, and we associate that perceptual feeling with the idea of hearing it. And we see something over and over again, and we associate that perception, that subjective feeling, with the idea of seeing it. And further, each of the systems, the visual or auditory system, has distinct wiring, so that there's not just a given set of neurons that's always wired the same way at all levels of our brain, but each of our different brain areas has a unique connections between and within areas. So one way that the brain represents the outside world is through a spatial representation. So the neocortex represents the sensory field of our vision, for instance, as a spatial map of what we're seeing, of an image. Another example is a topographic map of the body. So our somatosensory system has a representation of our entire body, and a given neuron in our somatosensory system might have a receptive field on your fingertip, whereas another neuron might have a receptive field on your forearm. And in this way, a topographic map of the body or an area of the sensory world can be created. In mammals like us, each sensory system has at least one primary cortical area in the outer cortex. We'll see the primary visual cortex today. And usually these project onto secondary areas, as we'll see today in the visual system. So we've learned then about this general idea about sensation and perception, where sensation is the physical aspect of the process. It's the registration of the stimulus by the sensory organ. It can be described perfectly by a textbook because it's a physical process that can be measured and observed. Whereas perception is something that is unobservable. It's our subjective interpretation of those sensations. Mine might be different from yours, for instance, and the way I see red, the way I perceive red might be completely different from the way you perceive red. Our visual experience, furthermore, isn't an objective reproduction. It's not a perfect copy of what's out there in the world, but it's a subjective construction that's manufactured by our brain. We're making it up as we go along. So we can see things that aren't really there, and we can interpret things in different ways at different times. You've all seen different visual illusions. These perceptual illusions play with that subjective nature of our visual experience. They are useful for studying the visual system because they are good examples where the sensation remains the same, but the perception changes. So it allows researchers to tease apart the sensation aspect and the perception aspect. You might even, as you look at these, have the two interpretations of the images flipping back and forth, indicating this flipping of your subjective experience given just a constant input. Furthermore, this can be modified by your top-down expectation. So if I tell you on the bottom that there's a cat facing the right here with its head on the right as well, you can interpret that image differently than if I told you something differently, indicating that some higher level parts of our brain can modify the activity of lower level parts, something we'll learn about throughout the course and in your future courses about neuroscience. Next, we'll talk in detail about the functional anatomy of the visual system, something that you likely have covered before. So vision as humans is our primary sensory experience. We have a large section of our brain dedicated to vision compared to any other sense. And by understanding the visual systems organization, we can therefore better understand the human brain and understand the other senses as well. Our visual system measures light from the electromagnetic spectrum. Light is the electromagnetic energy that we see. The range that's visible to humans is from about 400 nanometers to 700 nanometers, from violet to red, the colors we see at the edges of the rainbow. A nanometer is a billionth of a meter, and this is representing the wavelength that the particles are flying through space in. So when we see photons traveling into our eyes, vibrating at different wavelengths, our visual system perceives them as different colors. In fact, they're just photons that are vibrating at different wavelengths because of the way our photoreceptors differentially respond to different photons of the different energies. We can have perception of different colors. Other animals like butterflies and honeybees can detect ultraviolet light, and night vision devices use infrared light, like the police helicopter driving around at night. Okay, so some animals can see outside of this spectrum, and therefore they would have perceptions very different from ours when they looked at something that was illuminated by ultraviolet light. The eyeball itself, the physiology of the eyeball includes the cornea, which is this clear outer covering on the eye that we touch when we put on our contact lenses. The iris opens and closes and allows more or less light in. And the iris is the colored area of your eye. The hole in the middle of the iris is called the pupil. And the pupil can expand and contract to let more or less light in. The lens is a tissue behind your pupil that focuses the light, much like the lens on the front of your camera. And it allows for us to either look at something really close up or look at something really far away by changing the way that the light is bending as it travels through the front of our eye. The retina is on the back of the sphere of our eye. So all this stuff we've just talked about is on the front, and the retina is back here on the back. The light goes through the eyeball and hits the retina on the back of the eye, where it initiates neural activity. So we've talked about much of this stuff in the previous discussion on the previous slide. Here's an example showing the light coming in from bouncing off of a key in a room, entering through the sclera and through the cornea, in through the pupil, which is constricted by the iris, where it's focused by the lens such that it hits the back of the retina. Notice that it's flipped upside down on the back of the retina. And then notice on the back of the retina that leaving the back of the retina is the optic nerve and blood vessels. When I was talking to my son today about this, he said, oh yeah, that's what's dangling, where people's eyes are still dangling from when their eyes fall out of their head in scary movies. And he's right. Okay, so the back of this retina here is where all the photoreceptors are that are going to detect this light and send signals into the brain. Here's an image of the retina that your eye doctor can take that you might have seen, showing the fovea, which we'll talk about, an area where there's very dense packing of cones allowing us to see very clearly, as well as the blind spot. This optic disc here is a small hole in the back of the retina where the nerve and blood vessels leave and where there's no photoreceptors. One interesting thing to think about is that the lens can shift its size because there's small muscles here inside the eye. Not only are there muscles that move our eyes around in our head, and we'll see why, but there's muscles inside the eye that modify the lens, and which, as you know, can get tired from too much reading. So many of us suffer from one of these conditions, either nearsightedness or farsightedness. Myopia or nearsightedness is inability to see things far away. That's why I wear these glasses. And it brings, it occurs because the focal point of the light falls short of the back of the retina. Whereas hyperopia is a farsightedness, is when you can't read close up and you need reading glasses. This happens usually as we age most often. And this happens because the light falls beyond the retina. So here's an example, a diagram showing normal vision where the light is perfectly focused on the back of the retina versus my eyes that look like this, that have myopia from too much reading and not enough looking at far things away from myself. And hyperopia, as we age, our eyes become hyperopic and our lens can't accommodate as well. And the focal point is beyond the eye. So people often, as they age, need bifocals if they were myopic before, because their lens being unable to focus, they become hyperopic as well, and they have two lenses built into one set of glasses. The retina, as we mentioned, is a light-sensitive surface at the back of the eye. It is full of neurons and the photoreceptor cells, the rods and cones. These photoreceptor cells transduce light into action potentials. They discriminate different wavelengths because some of the cells are differentially activated by different colors. And the retina works in a wide range of light intensities because it has some cells that are sensitive to very, very faint amounts of light as well. The fovea, as we've seen, is a region at the very center of the retina that's specialized for high acuity vision. When we need to read, we need to very finely tell apart small details in our visual field. And so we need to use our fovea, and to do so, we need to move our eyes around the page because we can't read in our peripheral vision because it has much lower density of these cones. And the receptive field of the fovea is right where you're looking. So right where you're looking is the receptive field of the fovea. Here's an example of the fovea showing a couple of its features. One is that the rest of the cells that are usually in front of the rods and cones are moved off to the side, creating this small divot. And this allows light to directly hit these cones. You can also see that the cones are completely covering this area, whereas the rods are more present in the peripheral areas of the retina. And then we can see something we'll talk about in the coming slides. We can see the second and third cells in the pathway leading to the optic nerve. Here's a electron microscope image of a fovea as well. So because of our cone's density in the fovea and a relative sparsity in our peripheral vision, our vision is better in the center of our visual field. And as we move out into our periphery, for instance, we need larger and larger letters for us to be able to read them. Finally, we have a blind spot in our eyes, in each eye. This is the region of the retina where the axons forming the optic nerve leave the eye, and there's no photoreceptors there. And so if you close one eye and you hold a pencil just in the right spot, you won't be able to see the eraser. However, our visual system solves this problem by having the optic disc in a different location on each eye. So it's always lateral to the fovea. The fovea is in the center of our retina, and lateral, or this would be medial towards the center of my head, and lateral would be towards outside of my head. And therefore, one eye can fill in the image for the other eye. Our brain also does extra work to fill in images as well. And for instance, if you close one eye and look at a patterned wall, you can see the pattern completely. So your brain fills in that pattern in the missing area. The photoreceptor cells are rods and cones. Rods are more plentiful. They're sensitive to low levels of light at night, and they're used mainly for night vision because of this. And they only have one type of retinal pigment. That is that all rods are similar and that all respond to the same distribution of wavelengths. Cones, however, are more responsive to bright light. And they're specialized for color vision and high acuity because of their dense packing and because of their 3 different types of pigments. They're present mostly in the fovea, and as we'll see, they have these 3 different types. So here's the cones responsive to bright light and fine detail and color, and the rods, which have a different shape and morphology, you can see. A much longer outer segment, and in the outer segment is where these reactions between the light occur that lead to the signal being sent. Here's an image in an electron microscope showing the 2 types of cells. So the 3 types of cones absorb light over a range of frequencies. It's not just one particular wavelength of light, but across the range of frequencies, as we'll see on the next slide. They'll respond, but they have a peak absorption at a particular wavelength or color. So there's a blue, a green, and a red leaning cone. But you can see on the next slide, they all respond to most colors of light. They'll just fire the most for that particular color that they're sensitive to. There's about equal numbers of red and green, but fewer of these short wavelength cones. So here we can see the distribution of different wavelengths on the x-axis, and the response, or how many action potentials fire per second of those cones. And you can see that depending on the wavelength, the cone will have a different response, and the peak response for each of the 3 types of cones is different. There's a blue, a short cone, a medium cone around green, and a large cone, which we call the red cone, but you can see it's much more close to the orange area. The textbook mentions how on the X chromosome, it's encoded for these long cones, and that some women in our species have 2 types of these long cones, allowing them to have particularly effective. Some people in our species with 2 X chromosomes, therefore, sorry, not women, can have 2 of these long cones and therefore have particularly effective color vision in the red range. They could see more shades of red. So here are the receptors on the back of the retina. You can see that generally they're randomly spaced, so they'll be red, green, and blue cones in different places, and rods intermixed between them, although the rods are relatively sparse in the fovea. Well, on the previous slide, you can see that the rods also have a distribution of wavelengths that they respond to, but there's not different types. So that at night, when we look and see faint amounts of light coming in and let's activate these rods, we can't tell the colors apart because there's only one type of cell throughout the eye. When we zoom into the back of the retina and we take a slice through it, we can see 5 types of cells. We can see these rods and cones, and then 4 other cells along the pathway. Bipolar cells are the next step that the signals from the rods and cones get sent to. And from there, they get sent on to these retinal ganglion cells, which have an axon that leads the eye. Horizontal and amacrine cells are little interneurons in the connections between those 3 types of cells, as we'll see. So the horizontal cells are based on the connections between the photoreceptors and the bipolar cells, and the amacrine cells link together the bipolar and the ganglion cells. Here's a diagram. So here's the rods and the cones at the back of the eye. The light has to travel through all this mess of neurons, and generally because the cell membrane is clear, that's okay. So the light can go back here and hit these rods and cones. They send a signal to the bipolar cells, and the bipolar cells then send a signal on to the ganglion cells, the retinal ganglion cells in this case, and they have axons that lead. Here we can see the horizontal cells and the amacrine cells as interneurons in those pathways, which integrate information over multiple parts of the retina. The retinal ganglion cells are found to come in 2 types, magnocellular and parvocellular, based off both what type of input they receive from the rods and cones, and also where they send their projections to in the next stage of the visual pathway. So the magnocellular cells are larger, they receive input more from rods, and they're sensitive to light and movement. The parvocellular cells are smaller and receive information more from the cones, and they're sensitive therefore to color. Once information leaves the eyes, the axons form the optic nerve, and the optic nerve travels into the brain. It crosses over at this area called the optic chiasm, which we'll talk about in a couple slides. And from there, these axons project into an area in the thalamus, in the center of the brain, called the lateral geniculate nucleus, the LGN. The lateral geniculate nucleus. The lateral geniculate nucleus has 6 layers. The 2 innermost layers receive information from those magnocellular cells about light and motion. And the outermost layers, 3, 4, 5, and 6, receive information from the cones about color. The optic chiasm is the junction of the optic nerves from each eye. So some information from each eye stays in the same hemisphere and goes to the visual cortex, and some crosses over to the other hemisphere. Axons from the nasal half of the retina, so the retina closer to the nose, once they go into the head, so this nasal half of my retina will send information into my head and cross over to the contralateral side of my brain. Whereas the temporal hemiretina will go to that optic chiasm, but it will stay on the same side and end up on the ipsilateral, the same side of the brain from which it started. So information, because of this, information from the left visual field, everything over here to this side of me in this case, goes to the right side of each eyeball. So it goes to the right side of my left eyeball, the nasal side, and it goes to the right side of my right eyeball, the temporal side. And so the right side of each eyeball goes to the right hemisphere. So information from the left visual field always ends up in the right hemisphere based off these connective pathways. Here's a nice diagram showing it as well. So light that falls on the right half of each retina comes from the left side of the visual field. So here is that shown in red, this left visual field hitting the right side of each retina, traveling the temporal hemiretina of the right eye and the nasal hemiretina of the left eye, both combining their information and sending it into the lateral geniculate nucleus on the right side, and then on to the primary visual cortex on the right hemisphere. And so in this way, you can see that everything to the left of this individual ends up in their right visual field. There are 3 visual pathways in our brains as human mammals, and we'll talk mostly in this course and in future courses about human neuroscience about the geniculostriate pathway, which is what we just saw in the previous slide, the retina to the LGN to the visual cortex. However, you should know there are 2 other pathways. One is more prevalent, or is actually the visual pathway in reptiles and birds, the Tectopulvinar pathway, which we humans still have. The Tectopulvinar pathway are projections from the retina to the superior colliculus in our brainstem, and then to the thalamus and on to the parietal and temporal areas. And the Tectopulvinar pathway is really important for quickly locating things in our visual world and reaching out and grabbing them or interacting with them. So if anyone's ever thrown a ball at you and said, catch, and you reach out and grab it, you're using your Tectopulvinar system, much like a frog with a fly buzzing around it needs to shoot out its tongue and grab it. It's using the superior colliculus pathway. Finally, the retino-hypothalamic tract is a 3rd pathway in our visual system that is relevant for this discussion we just had in Alberta about daylight savings time. So the light from the sun enters our eyes every day and not only informs us about all the stuff we're seeing about the world, but also creates a circadian rhythm in our brain so that our brain knows when daytime and nighttime is. And it doesn't do this just by activating the visual cortex in these other two pathways. There's specific retinal ganglion cells that are sensitive to light, that even if someone's blind can still be activated. And these retinal ganglion cells synapse in the hypothalamus, an area called the suprachiasmatic nucleus. And in this case, they have a role in regulating our circadian rhythms. So we need this early morning light that we get from changing our clocks in a couple weeks. And this early morning light starts our circadian rhythm each day, keeping our brain in synchrony with the rhythm of the day. So here's the two of those main visual pathways, the lateral, the geniculate striate system, which is most used in humans, traveling to the striate cortex, and then on to other visual areas. The striate cortex is another name for the primary visual area. And then the tectopulvinar system, which goes directly into the superior colliculus, into the thalamus, and then on to these other cortical areas. So it bypasses the main striate cortex pathway, the primary visual cortex. And we're often not aware of activity that's happening in this pathway, so it can happen unconsciously. So now we'll talk mostly for the rest of today about the geniculostriate system, I believe. So the primary visual cortex, or another name for it, is the striate cortex because of the way it looks in this stain on the microscope. And the primary visual cortex is the area that these axons that leave the lateral geniculate nucleus, this is where they synapse. And from here, the two pathways emerge in the visual system, one going up to the parietal lobe and one going down to the temporal lobe. And we'll see what those two pathways do and how they process different parts of our visual world. So the dorsal visual pathway starts back here in the primary visual cortex and goes up to the parietal lobe. This is a pathway related to how we do things when we want to reach out and grab something and interact with it. Alternatively, this ventral pathway originates in the visual cortex and then projects to the temporal cortex and helps us identify the objects that we're looking at. So I can look and notice which kind of drink that I have. So here are those two dorsal and ventral streams, which we'll talk about more in this lecture and other times in the class as well. So one thing we didn't mention about the lateral geniculate nucleus is that it receives information from each eye. And information from the left eye and the right eye are kept separately. So the right LGN, even though it gets information from the right half of one retina and the right half of the other retina, still keeps those two sets of overlapping information separate. Right? Our eyes both look forward and they have slightly overlapping views of the world. And by separating out the layers in which those information go, the LGN can separate out the information from the ipsilateral and the contralateral retina. Furthermore, we saw that the magnocellular and parvocellular cells of the retina project to different layers of the thalamus here. And so you can see which layers, which types of cells project to. So these P and M retinal ganglion cells send separate pathways to the thalamus, and even the segregation of information from the left and right eye and from the P and M pathway continues in the primary visual cortex. So the signal from our visual system is kind of being chunked up into different features and kept separate in the system. Which eye did this come from? Was it moving? Was it colorful? So the left and the right eyes are sending separate and related images to the thalamus. So here we can see the right half of each eye, the nasal left eye and the temporal right eye, both projecting to the right LGN and projecting to different layers of the LGN as well. Once we get into the cortex, there's a new type of organization beyond just the layers of the LGN. There's something called a cortical column. So this dinner napkin typed cortex that's folded together to fit into our skull, each little section of it you can think of as a tiny, like a Lego brick piece, each of those little towers of the Lego brick. And each of those columns of the brain, because it has 6 layers, we call it a column. Remember that the brain has 6 layers of cells. It's about 0.5 millimeter square. So you can picture these tiny little 0.5 millimeter functional units of the brain. And therefore the brain is kind of like a mosaic of these cortical columns. And they go up and down perpendicular to the surface of the brain. So from each LGN nucleus, information from the retina is sent to adjacent cortical columns. So if there was a signal in adjacent parts of the LGN, now there's going to be a signal in adjacent parts of the cortex. And this maintains the spatial information from the retina. So the different cells, 2 cells right beside each other in the retina will send their signals to 2 cells right beside each other in the LGN, and those will send their signals to 2 cells right beside each other in the primary visual cortex. So M cells in layers 1 and 2 and P cells in layers 3 through 6 actually project both to layer 4 in the primary visual cortex, but to different sublayers of layer 4, keeping information separate. Furthermore, the left and right eye information projects to adjacent columns in the visual cortex. And if you stain the visual cortex or look at it through a light microscope while the animal's alive, you can see these ocular dominance columns alternating information from the left and the right eye. The main overall message here is this continued segregation of the different information, chunking of the different types of information into different circuits. So it all comes in and then gets separated out into different features. The visual cortex has after this a number of different regions that process information in different ways. The primary visual cortex we've seen gets information from the LGN, but there's V2, V3, V4, and V5, and also extrastriate cortex beyond the primary visual area region, which process visual information from V1 as it moves through the visual system. If you look either in a medial or a lateral view of the brain, you can see that these visual areas are tightly packed together at the back of the brain here, but they're separate from one another. And information will be shared between them as it gets moved through the system. As cells in V1 fire, they'll synapse onto cells in V2 and then activate them and make them more likely to fire. If we look at a microscope at a slice of V1, we see in the microscope blobs, small regions that contain color-sensitive neurons, and interblobs, or regions that separate these blobs. And so the blobs are where the color information from our parvocellular pathway is ending up, and the interblob regions has more information from the magnocellular pathway about form and motion. So here's the slice of V1 and V2 showing these blobs and interblobs in V1, separating out the color and motion information. And then in V2, another feature is these stripes, which come in both thin and thick widths. and the pale zones in between them. And we'll see that the different types of visual information ends up flowing through these different parts of V2, much like different parts of a subway system. So neurons in the blobs take part in color perception. So information about color, form, and motion continues to be separated into V2. The thick stripes receive movement information, the thin stripes receive color-sensitive information, and the pale zones in between them receive form-sensitive information. This kind of thing is much easier for you because it's an open book exam. You don't have to remember these associations that seem pretty random. So here's an overall diagram showing information from the LGN, which already had the M and P pathways separate, coming into different parts of the V1, blobs containing color information, and the inner blobs containing form and movement information. These project to different parts of V2, form to the into the pale zones between the thin and thick stripes, and movement information going to the thick stripes. The color information is contained in these thin stripes. We'll see then that the information continues to be separated such that form and motion end up traveling on predominantly to the parietal lobe and the dorsal stream, and moving forms and color end up predominantly being sent in the ventral stream to the temporal lobe. So this information differentially allows us to grab things out in the world and reach and interact with them, or identify what things are based off the different types of visual information in the signals. But one other curious peculiarity of the brain that remains a mystery is how these separate bits of information, the color and the motion and the shape of an object, say like a fire truck that's racing on the street in front of us, even though they're processed in separate regions, it's unclear how all these different perceptions that we have are bound together into one object. This is called the binding problem. There's not one area beyond the parietal and temporal lobe where all the information arrives, and that's our consciousness. We're conscious of things in these different pathways as if they're bound together into objects, even though their information is separate in different pathways of the brain. Beyond the occipital cortex, we have a complicated set of visual areas that process different aspects of the visual regions in the ventral stream. Sorry, different features and types of objects like faces and scenes in the ventral stream. So here's two areas on the bottom side of the cortex, a picture of the brain that's been a 3D model of the brain that's been inflated up so that there's no grooves in it anymore, showing this fusiform face area and parahippocampal place area, areas that are differentially active, that is, a lot of blood flows to them when we see those types of visual scenes. Also, people with damage to these areas seem to have disorders associated with seeing those types of objects. In the dorsal pathway, there's three other visual areas here: the lateral interparietal, anterior interparietal, and the parietal reach region. These are just three of the many other visual areas. These are involved in eye movements to interesting things that we need to see, or reaching out and grabbing something. And I've already mentioned that these damage to the FFA can lead to prosopagnosia, or an inability to recognize faces. Okay, so next we'll talk about the how the visual system encodes the location of things in the world. I need another drink. So we keep talking about the visual field. The visual field is the entire world that we can see with our eyeballs. It's divided into the left and the right side, and information from the left side goes to the right hemisphere, information from the right side goes to the left hemisphere. That's where we are so far. And so you can picture staring at a screen that completely illuminates your vision with a cross in the middle. Everything to the right of you in blue would travel to your left hemisphere. Everything on the left of you in red will travel to your right hemisphere. Each of the retinal ganglion cells, as we saw, has a tiny receptive field. It only responds to stimulation of a small circular patch of the retina. So that's the retinal ganglion cell's receptive field. That's where light can hit the retina that will make that retinal ganglion cell fire. And it represents a small circular part of the world as well. And so that retinal ganglion cell can encode the location of something in the world by whether it's firing or not. If it's firing, then there was something happening in that small section of the visual field. If it wasn't firing, then there wasn't something occurring in that small section of the visual field. And a corollary of that is that light falling on a different part of the visual field will activate a different retinal ganglion cell. So as we move up into the LGN, and when I say up, I mean up the visual hierarchy that processes more and more complex parts of the visual scene. And these LGN neurons also have receptive fields. So each LGN neuron represents some particular part of our visual space. And these go on to project to the primary visual cortex V1, creating this topographic map, which we can call a retinotopic map. There's a map of the image of the retina that's continued throughout the visual system. But as we move into the visual cortex, the receptive fields of the cells there gets larger than those in the eyeball. So a given neuron in our primary visual cortex might respond to a larger section of the visual field than a given retinal ganglion cell in the retina, because it's combining the information from multiple retinal ganglion cells. Another way you can think about this like hierarchy. And because we have so many cells in our fovea, and they all send their axons to the LGN, and they all send axons to the primary visual cortex, we end up with a large section of the primary visual cortex dedicated to receiving all that foveal information, and smaller sections of the cortex dedicated to receiving information from the periphery. So the visual cortex is like morphed or stretched, and the part of our retina that with lots of sensors has a much larger amount of real estate there. So one can also see that this retinotopic information is held throughout the pathways, in part because of the way that adjacent sections of the retina project to adjacent sections of the LGN and adjacent layers of the LGN. So the spatial map on the retina is kept consistent inside the LGN. Another way to think about it is that there's like a picture of the image that hits the back of your retina in the LGN. The LGN has a picture of it, and then the primary visual cortex has a picture of it. Not a picture, but a informational representation of that image. But it gets morphed in different ways as it moves through the visual system. It becomes less like reality. And okay, so the foveal vision, the vision of what we're looking right at, is represented at the very back, the occipital pole, it's called. And the periphery is represented more anteriorly. So in areas further away from the very back of the brain. The upper part of the visual field is represented in the lower half of V1, and the lower part is represented in the upper half of V1. And as we move into V2, V3, V4, and V5, there's similar topographic maps or retinotopic maps. There's still pictures of what hit the retina. As we saw, the fovea sends information to a disproportionately large part of the occipital cortex because of the high acuity and number of cells there. So this small section of our visual field above and to the left of where we're looking has this large section of the primary visual cortex dedicated to processing, whereas this peripheral area, which is much larger in our visual field, has a smaller section of the cortex because it has fewer cells synapsing onto fewer cells there. One can see here that the upper visual field is represented in the lower half, unless you're colorblind, and then you might not be able to tell apart these two colors because you might have only two types of cones. 5% of male humans have this issue. And what else? Oh, and the left visual field is represented in the right visual cortex here. Because the receptive fields of the cortical neurons are composed of many retinal ganglion cells, more than one retinal ganglion cells synapses onto that neuron, their receptive field is larger. And so the receptive field actually grows as we move up the hierarchy because a couple cells all synapse onto this LGN cell, and then all these LGN cells all synapse onto this V1 cell. And so this V1 cell will fire not just when something occurs in this small section of the retinal ganglion cell receptive field, but if anything in this area gets activated. In between our two hemispheres, we remember there's a corpus callosum, a white matter tract of axons connecting the hemispheres together. Only certain parts of the brain are connected between the hemispheres. The frontal lobes have very tight communication between the left and the right hemisphere, but the occipital lobes have almost none, except the cells that represent information about the midline or right above and below where we're looking. They have connections over the corpus callosum, which allows their receptive fields to overlap. And so you can represent things across your visual field, left and right side of your visual field. Here's a monkey's brain showing a little bit different anatomy, but you can see they're showing the occipital lobe having no connections. Okay, next we're going to talk about the neural activity of cells in the visual system. This is really important work that won the Nobel Prize a couple decades ago for the understanding of what the cells of the visual system did and how they represented the world by Hubel and Wiesel in New York. So the visual system, as we'll see, responds to different features of the image. So one way we can test this is by placing different types of stimuli in front of people or animals and seeing which parts of their brain are active. So we can place a straight line, just a line on a piece of paper in front of the eye. And if the cell that you're measuring from likes that line, it will increase or decrease its firing. If it responds to that line at all, it will decrease or increase its firing. And it might increase or decrease. If it has no reaction, then it doesn't care about that line at all. So if it cares and it changes its firing rate, it's generating information about the line. And what we'll find is that, say, as we change the orientation of that line, so that we go from a vertical to a horizontal or somewhere in between, an example is that the cell representing that stimulus with a receptive field in that location might change its firing rate. So it might care more about vertical lines than horizontal lines. Just like neurons are selective for particular locations in the world, neurons are selective for particular features about the stimulus. Some other cells might care more about red than green, for instance. So as we know, though, all we can do is record the neuron's firing rate, and we can see if it's excited or inhibited by a particular stimulus. The retinal ganglion cells, if you record from them and present different stimuli to the eye, they only respond to the presence or absence of light, not to any particular shape. And they have a particular arrangement of circular receptive fields called an on-center or an off-surround, where the center of the receptive field and the periphery of the receptive field have differential effects if light hits them. So these on-center cells are excited when light falls in the center part of the receptive field, and they're inhibited when light falls in the surrounding part. So if you shine light across the whole receptive field, they'll only fire weakly. I will see a picture of that on the next slide. There's the opposite, there's off-center cells that when they're excited when light falls on the surround, but not when it falls on the middle. And so these retinal ganglion cells, they are not as simple as we thought. They don't just respond to light being present in some part of the world. They respond to the contrast of light being surrounded by darkness. And it's this contrast that they care about. So here you can see the cell fires the most when light hits its middle and there's darkness around its edge. It gets inhibited when light hits its inhibitory surround area. And conversely, the off-center cell has the opposite effect. If you were to shine light completely on the on and off part of this cell on the left, the on-center cell, it would fire an intermediary amount because it would be activated by the firing in the middle, by the light in the middle, but it'd be also inhibited by this light hitting the outside. So adjacent retinal ganglion cells receive input from overlapping sets of photoreceptors. So they're, as we saw, the receptive fields overlap. So if you shine a small spot of light on the retina, it will hit some of the on and some of the off center of many of these cells at once. Let's see what that looks like. Right, so you have all these cells with all these overlapping receptive fields, and two of them have almost overlapping receptive fields, such that there's like a sweet spot that would make both those neurons fire. And this can help us picture how the neurons would come to represent something like a line, because if enough of these were all overlapping together here in a line and a line hit them, then they would all be active, and that would tell the next step of the visual system that this line was present. So the retinal ganglion cells don't just care about luminance, the amount of light, but the contrast of luminance, how much light surrounded by darkness. So that the visual system can start to identify different shapes. Here's one example of how an edge between light and dark, when we look at it, different retinal ganglion cells with receptive fields either completely in the dark or completely in the light or halfway between will have differential firing. And by combining the information about different firing of these retinal ganglion cells, the brain can represent where this edge is. A cell completely in the dark will have a baseline firing rate. A cell completely in the light might have a slightly increased firing rate to that. One half in the dark and half in the light might be equally inhibited and disinhibited. Whereas you can see that this light just firing on the inhibitory surround of this cell will cause it to be inhibited compared to baseline. And light firing on the on center with some of the off surround not illuminated by light causes this D cell to fire a great deal. And the edge can therefore be understood as the inflection point here of this line across this baseline rate. And our perception can then build this idea of an edge out of that information. So these V1 neurons receive information from multiple retinal ganglion cells as well. And so they're going to receive information from multiple of these on-center off-surround cells. They have a much larger receptive field. And they start to behave more like orientation detectors, as I alluded to. Detectors of the orientation of different lines. And first we'll talk about simple cells, and then we'll talk about complex cells. They're both in V1 and they have slightly different receptive field properties. Simple cells have rectangular receptive fields with an on middle and an off surround. So they love things like this with light surrounded by darkness. And they'll also respond more at particular orientations. Okay, so if the on gets shone with light and there's no light in the off, they'll have a strong response. If light is shone across the three, there won't be a response. However, there might be a separate neuron that prefers a different orientation of light and will therefore fire much more strongly to that second condition of light being illuminated at a different angle. Okay, so if you're just listening to one of these neurons firing and you move the stimulus around like this, there'll be a particular time where it will fire the most at the orientation that it prefers. Complex cells not only have this kind of bar of light of a particular orientation that makes them fire, but it needs to be moving. And so there needs to be light of a particular orientation moving through the visual field to get them to fire. And they'll fire less strongly to light of a different orientation moving. Finally, there's something called hypercomplex cells. They have an inhibitory area at one side. So they are just like those complex cells, but once the moving object gets to a particular area in the receptive field, they'll stop firing. You could imagine these being useful to as the visual system learns about edges and things getting occluded behind, something like that. So it seems like we're missing some level of explanation of how all these different receptive fields that overlap from all the different retinal ganglion cells can come to represent something like a bar of light. So input to any given V1 neuron comes from a whole group of retinal ganglion cells. And one particular V1 neuron might be connected to a group of ganglion cells that together form a row. So whenever that whole row of retinal ganglion cells gets activated, that V1 neuron will fire. But if different retinal ganglion cells are activated, that V1 neuron won't fire. So here that is in an image. So imagine a particular V1 neuron that happens to be connected to these 7 retinal ganglion cells. It will fire strongly when a bar of light shines across them this way, but it will fire less strongly when a bar of light is oriented slightly differently. But we also have to picture another V1 neuron that is excitatorily connected to a different row of neurons, maybe this one, this one, this one, this one, this one, and this one. And that neuron will fire more. It will represent that new orientation of line more than this one we're seeing on the slide now. And throughout the millions of these V1 neurons, all randomly connected to different groups of these retinal ganglion cells, they can represent different orientations of lines in this way. As we saw, these neurons are organized in a column. So all the neurons up and down the column, the 6 layers of the cortical column, all seem to have similar functions. So if you recorded from any of those neurons, they would all become activated by that vertical line if one of them was. So all the inputs come to layer 4 and then connect vertically to the other cells, creating this coordinated activity. And we saw the ocular dominance columns, which receive information from the left or the right eye separately. Here's one of these cortical columns. It might have made more sense to talk about this a couple of minutes ago when we were talking about the cortical columns originally. But we'll see why we want to talk about them again here, because the way we can think about them as functional units. This whole group of cells will all process vertical lines, or will all process the color red or a face. So adjacent columns would house neurons that, because of their differential connections to the previous area, will become activated by slightly different orientations of lines. And adjacent columns on the other direction on the cortex might be activated by the opposite eye, so left and right eye ocular dominance columns. As you move up and down the 6 layers, for some reason here shown as 5 layers, all the neurons in those layers have the same feature that they care about. As we move up into the temporal cortex and we look at what the various cells do, we find that they become excited by more complex visual stimuli like hands or faces or letters. And some might be selective to like this orientation of face, some to a turned face or a sideways face, some to the different rotations of the head or to certain facial expressions. And they might even fire to the same face, but from different angles, allowing for something like stimulus equivalence, where we can recognize something like a banana, even in very different visual circumstances. In the temporal lobe, it was found that pretty complex features were needed. So you need a combination of lines of particular orientations and colors and different textures. So these neurons respond to very complex groups of stimuli. And if you look up and down a column in this area, you might find kind of similar representation across the 6 layers, but with maybe slightly different activation, so that different neurons in one column might respond to different types of facial expressions or different shapes, all similar in their amount of lines. And so you can see, instead of just basic features like the orientation of a line, now there's kind of higher level categories like hands or faces or fruits represented by these higher level visual neurons. In V1, the preference of neurons is pretty fixed, doesn't change throughout our lifetime, but in the temporal lobe, it can change with experience, indicating the more flexible nature of these representations here and how they can be modified with learning. Finally, how our visual system processes color is important to understand. And so when we think about physical mixing of colors, we can think about subtractive color mixing like paints, where you can represent the entire range of colors by mixing 3 paint colors, red, blue, and yellow. And if you combine all 3 of them, you'll make black. And if you don't use any of them, you'll have white. And this is the same property of the cones in our retina. Because they have 3 different sensitivities, they can combine their information together to represent any different colors in the spectrum. Alternatively, additive color mixing is like light mixing together. When you have no colors mixing, you have darkness, and when you have all three colors mixing, you have white. And so a white surface has all of the colors built into it. So this subtractive color mixing relates to this theory of color vision called the trichromatic color vision theory. It explains color vision as being primarily the relative activation of the red, blue, and green cones. And we see color by combining these relative responses to understand what color we're seeing. And this explains why people with only two of these cones have problems in detecting the full range of colors that other people can. However, from our basic understanding of the colors space, we know that there's actually four basic colors. There's red, green, yellow, and blue. And there's this interesting feature called after images, where if you stare at a colorful image for a while and you present a white screen after, you get the opposite colors in this perception of an after image of the same image. So an alternative theory of color vision is called the opponent process theory. And it's likely that in the very early stages in the retina, this trichromatic activation happens. We combine the relative activity of the three cones to understand what color is present. But then at higher levels, like the retinal ganglion cells, opponent processing is happening. So because of differential connections to different cones, retinal ganglion cells might come to represent how red versus green something is, or how blue versus yellow something is. And this can even occur in the on-off center surround type of way. So about 60% of the retinal ganglion cells, I'll show you the next slide first, respond to, say, red light in the middle, and they'll respond to green light around the outside. They'll respond best to red light surrounded by green light. I think of these as like raspberry detecting retinal ganglion cells that are really good for detecting the contrast between red and green. This is the demonstration of that opponent processing. You could stare at the red and blue image for a couple minutes without moving your eyes or head. And then you can move over to the other image and you should see yellow where there was blue and green where there was red, indicating what you might think of as like a fatigue of the neurons that were representing red. They were saying red, 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 red. And then when the red goes away, they're kind of exhausted. And so they'll come to represent this white screen as being green. So we saw from microscope images that these color sensitive neurons are kind of intermixed between these orientation and ocular dominance columns, and they're represented in these blobs. So there's color sensitive cells found in the blobs of V1, which also have opponent processing. So that they'll represent how red versus green, or how blue versus yellow, or how black versus white something was. Finally, this information, sorry, not finally, but next this information moves about color moves to the cortical area V4. V4 doesn't respond to the wavelengths of light hitting the retina, but responds to the perceived color that you're seeing. So if you see a visual illusion where you see red, even though there's no red present, the V4 neurons that represent red will be active. So here's a really good indication of a difference between our perception and our sensation. The sensation of the early neurons will represent the exact wavelength of light hitting the retina, whereas the perception has a different kind of tint that's been added by our visual system. And these have the on-off inhibition as well. And the fact that they respond to the perceived color and not the veridical color indicates that they might be important for color constancy. This idea, for instance, that a banana, if you have a banana at sunset or at dusk or in a room inside or outside, it always appears yellow, even though the actual wavelengths of light bouncing off it and hitting your eye can be different in those different situations. Okay, what about in the dorsal stream? So that was showing the ventral stream and this importance for color processing and how the neurons there might represent color. In the dorsal stream, the parietal cortex is involved in action-related vision, or how we reach out and grab stuff. And these neurons, when you show someone sitting in a chair not doing anything or under anesthetic so they can't move, you show them visual information, these neurons don't fire. They only fire when there's an ability to move towards the object. And so some of these cells might process the kind of the graspability of an object, and they might even fire when an animal watches another animal, like yours might fire when you watch me reach out and grab this pen. Okay, the final thing we'll talk about is these kind of high-level visual representations and also some damage to the brain and what it tells us about, to our visual system and what it can tell us about the organization. Monocular blindness is destruction of one eye. So you lose sight in one of your eyes, either from a retina being damaged or the optic nerve. And this is very different from damage anywhere else in the visual system, in which your visual field gets damaged, not just one of your eyes. If you get damaged in just one eye, the other eye can compensate and you still have this full view of the world. However, if you get damage to your right visual primary cortex, for instance, it represents everything about the left side of space. And so you'll end up with something called a scotoma, or a blind spot, or a hemi or quadranopia, a blindness in a quadrant or half of the visual field. A scotoma is just a small version of these larger blindnesses that can take up a half or a quarter of the visual field. And scotomas can sometimes also occur prior to migraines. So here is an example of the type of injury that could lead to a hemianopia. The entire left hemisphere might be damaged in the visual cortex, or maybe only the lower half might be damaged, so only the right upper part of the visual field, or only a small part, so a tiny scotoma will be present in the visual field. And the textbook talks about these occurring before migraines as well, something that's happened to me a couple times. When we get damage to the what pathway, we end up with disorders called agnosias, or lack of knowing. These can be an inability to recognize objects or drawings of objects, inability to recognize colors, or prosopagnosia, one we've already learned about, an inability to recognize faces. Even though these patients can't recognize objects like an apple or a cup, they might still be able to shape their hands appropriately when they reach out and grab them, showing the differential activity of the dorsal stream and the ventral stream, the dorsal stream still being functioning while their ventral stream is not. So here's an example of a patient, JW, with an agnosia that can't copy simple shapes that are shown to him. So even though they can't identify objects or estimate their size, they can often appropriately reach out and grab things with the right grasp. And they might not even be aware of themselves doing that. Alternatively, if you get damage to the dorsal stream, but not the ventral stream, you can end up with disorders called ataxias. So this is an inability to use your vision to reach out and grab stuff and make other movements like jump into the right place. This occurs due to damage to the parietal cortex. And when this happens, one can still usually recognize objects using the alternative pathway. One good demonstration of these two pathways occurred across these two patients, DF, who had a ventral stream deficit, and RV, who had a dorsal stream deficit. So DF, even though they couldn't identify these different shapes because of their damaged ventral stream, when asked to grab them, reached out and grabbed them in the appropriate places given their shape at the long and other axes. Similar to how control participants did with normally functioning brains. However, RV, who had a dorsal stream deficit and was able to identify the different shapes, when told to reach out and grab them, picked them up in completely random places and made grasps that were inappropriate for the shape. Okay, so here's a final diagram showing the information from V1 going to V2 and then also on to V5 and V3. The pathway to V5 and V3 ends up at the parietal areas, taking that magnocellular information from the retina, kept separate in V1 in those different areas, onto the dorsal stream to represent information about motion and form. And then V1 also sends information to V2 and on to V3 and V4 about the dynamic form of objects and their color, leading to the input into these temporal visual areas. One important aspect that's shown here is that there's intercommunication at all levels of this hierarchy. So that's everything about the visual system for today. We've talked about general overview of sensation and perception. We talked about the anatomy itself, how the brain represents location in the visual world. We zoomed in on the neurons, saw how they fire depending on different types of visual stimuli. And then we talked about how damage to these different high-level brain areas can lead to differential effects on our vision, depending on if it's damage to the how pathway or the what pathway. And next lecture, we'll be talking about the auditory system. Good luck with your quiz.